Okay, we're going to go ahead and get started. It looks like our numbers have stabilized. So welcome to the third session of the SynCell 2021 speaker series, where we have some excellent speakers lined up for you. My name is Jacqueline Delora. I'm a postdoc in the Spatz Lab of the Cellular Biophysics Department at the Max Planck Institute for Medical Research in a sunny Stuttgart, Germany today. So on the schedule is our featured speaker, Dr. CLYDE HUTCHISON III, followed by a contributed talk by Dr. Oscar Stauffer. We encourage the audience to submit your questions through the Zoom webinar Q&A button that you find at the bottom of your screen, or if you are watching on YouTube Live, you can use the comments section there. So to begin, we are thrilled to have Professor CLYDE HUTCHISON III, who is a distinguished professor at the J. Craig Venter Institute in La Jolla, California, where he is a member of the Synthetic Biology Group, as well as a consultant for the company Synthetic Genomics Incorporated. Professor Hutchinson graduated from Yale University in 1960 with a Bachelor of Science in Physics. He then pursued his graduate studies at Caltech in the lab of Robert L. Sinsheimer and finished his Ph.D. in 1968. Professor Hutchinson then went on to join the faculty of the University of North Carolina at Chapel Hill from 1968 to 2005, where he now holds the title of Kenan Professor Emeritus. His work has focused on the molecular genetics of bacteriophage, bacteria, and mammals. Notably, during 1975 to 1976 in Fred Sanger's lab, he helped determine the first complete sequence of a DNA molecule. Then in 1978, he developed site-directed mutagenesis with Michael Smith. In 1990, he began work on mycoplasmas, as well as models for the minimal living cell, leading to collaboration with both Smith and Venter, and his current work on synthetic genomics. Currently, his real-time research priorities include understanding the minimal requirements for life, developing methods for genome design, and modeling the minimal synthetic cell with predictive computer models. We are really happy to have Professor Hutchinson, who will tell us about the essential universal tasks for a minimal living cell for the next 30 minutes, followed by 10 minutes of questions from the audience. Professor Hutchinson, you may now take over the screen and begin your presentation.
Okay, I think I'm taking over the screen. No, wrong thing. How do I get out of here? I want to start over.
Let's see. Sure. Let me take over one more time and then you can try again. How about from there?
Okay. When I press share screen, it says this will stop other share. Okay.
That's okay. Yeah, that's how you will override me.
Yeah. And this is the right one. Share. No, it isn't. Just a second, I'll be okay. There.
We have it.
Okay,
Perfect.
well, I'm
Thank
glad to be here. I appreciate being asked. This is something new for me. As you can probably tell, I'm not an experienced speaker on Zoom. And I want to give an update. I'm representing the synthetic biology group at the Venter Institute, and I want to give an update on our work with minimal living cell. And I'm going to be a bit heavy on the background. These pictures on the right are taking up a lot of space, by the way. In 1943, Erwin Schrödinger, who is most famous for his work in the origins of quantum mechanics, published a book about biology, and it's based on a series of lectures he gave in Dublin in 1943, and the book was published the next year, titled What is Life? And in this book, he poses the question, let's see, can I move this? There. I want to move that out of the way. Can you? Okay, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? So he wanted to be able to explain life in terms of physics and chemistry, which is logical since he was a physicist. And so he went on to speculate that the genetic information in an organism is contained in the chemical structure of something he referred to as an aperiodic crystal. Slide isn't advancing when I...
You can try using the arrows on the bottom left corner of your slide. If you click on one of yes.
Okay.
That will take you back. That one will take you forward.
Okay. So he proposed that there was this aperiodic crystal that contained the genetic information. And so when Watson and Crick started working on the structure of DNA, they had actually been influenced by his book. And here's a letter from Crick to Schrodinger, I guess trying to publicize their work on the DNA structure. And he says, Watson and I were discussing how we came to enter the field of molecular biology. We thought you might be interested in the enclosed reprints and think your term aperiodic crystal is going to be a very apt one. And in the paper, they went on to talk about information in the DNA. They said the sequence of bases on a single chain does not appear to be restricted in any way, thereby leaving up all possibilities for ways to encode information in the sequence of the molecule. So nowadays, we know how to determine DNA sequences. And if we view this question, what is life, through the lens of genomics, it becomes the question, what genes are necessary for life, what are their functions, and how do they work together to produce life? And in science in general, a lot of mileage has come out of the idea of thinking simply or looking for simple model systems. So the physicists had the hydrogen atom, and molecular biologists have simple systems like the phage Phi-X 174 that I helped sequence in 1975. And the mycoplasmas like M. genitalium, which has the smallest naturally occurring genome of a cell, at less than just about 500 genes. So these are simple systems. It's interesting, the reason the mycoplasmas are simple is the opposite reason from why hydrogen is simple. Hydrogen is simple because it's primitive, and hydrogen atoms can fuse in supernovae to produce larger atoms. On the other hand, the mycoplasmas are simple because they're minimal. They have descended from organisms with larger genomes, and material has been lost from the genome in the very permissive environment in which the microplasmas grow in organisms. So we're interested in synthetic biology, which postulates that the DNA genome is a piece of software and the cell expresses that with the other items in the cell. So we have, this is not a picture, it's a Venn diagram. At the Venner Institute, we've been interested in building a synthetic minimal cell that has only the core functions necessary for life. And a bacterial gene set contains functions which are essential for life, and also adaptive functions that provide other capabilities for the organism, like enable the bacterium to grow in a variety of environments. So our vision is, when we started this, and still is, a cell in which every gene is essential, and we know the biochemical function and biological role of every gene product. So why is synthetic cell? As another physicist, Richard Feynman, famously said, what I cannot create, I do not understand, which of course doesn't mean you understand everything you can create. And the other motivations for making a synthetic cell are as a proof of principle for moving designs to a living system. And it has required building a powerful set of tools for constructing and installing genomes. And also, it allows us to define the minimal set of genes required for life. So there are two ways to build reduced genomes. The more classical way is on the right, top down, delete non-essential parts of the genome sequentially till you get a smaller genome. And what we're doing here is on the left, here called bottom up. So we design a genome, build it by chemical synthesis, assemble it enzymatically, and then install it into a cell. And so hopefully someday soon, it will be possible to build cells from non-living components and put them together. But this is still a difficult problem, although I'm optimistic that progress will be made in this area. So what we did instead was to build a synthetic genome and install it into a living cell, producing a cell controlled by the synthetic genome, which we call a synthetic cell. And why do we feel justified in calling that a synthetic cell? Here's a Gedanken experiment that explains why. So if we assembled the cell from parts, as shown on the left, the genome is blue and all the other parts here are shown as green. And if we let that replicate, then eventually all the parts of the cell will have to be those specified by the blue genome, and we'll have what everyone would agree is a synthetic cell. On the right, if we introduce a synthetic genome into a recipient cell, which is a different genome, shown here in red, then if cell division goes on, we will eventually get a synthetic cell. And that the point is, in the cell where the genome's been transplanted, there aren't many protein molecules. There's only less than 1 million protein molecules. So on cell division, after 30 doublings, we'll have 10 to the 9th cells, and only one cell in 1,000 could have even a single molecule of protein from the original cell. So the whole cell now is made from components that are specified by the synthetic genome. So what cell did we want to minimize? We started from a mycoplasma named M. mycoides, and we started from a synthetic version of the wild type organism with just a few markers added to it. And this has a 1 megabase genome and encodes about 900 genes. It's a simple cell. It's a parasite on a mammalian host in nature, but in the laboratory can be grown in pure culture in a very rich medium. And it has a doubling time of about an hour and makes visible colonies in a couple of days. So we developed this design-build-test cycle for bacterial genomes. And really wish that those things weren't on the right of the screen. The design allows us to test, the design is based on gene essentiality, which is judged mainly by transposon bombardment of the genome and mapping by sequence the location of every place that a transposon can insert. And then we synthesize a set of reusable cassettes and subassemblies and build these into segments that are 1/8 of the genome. And we can now mix and match between wild type 1/8 genomes and reduced 1/8 genomes. These genomes can be assembled by recombination in yeast, and they can be propagated in yeast, whether they're viable bacterial genomes or not. They don't produce anything that's harmful to the yeast cells, so they can be carried along in the yeast cell as an extra chromosome. And then this genome is isolated, transplanted into bacteria and grown out. And then we can do things like transposon bombard again this and look and see whether inclusion of segments with deleted genomes has made any genes become essential that were non-essential before. And this isn't new stuff, so to shortcut a lot of work, I'll just say that we went through 3 cycles, reduced genome design 1, 2, and 3. And the first 2 did not produce any viable cells, but they did produce 1/8 genome segments that could be recombined with wild type segments to produce viable cells. So with our first design, all of the 8 segments worked in the context of the other 7 being wild type, but we could combine as many as 4 or 5. And by then, playing with the design, we went to design 2, and these produced 7/8 of the genome could be combined. And then when we diddled around with the 8th one to get it working, we were able to derive a viable cell, which is smaller than the smallest known natural genome. And this is called JCVI Syn 2.0. And then finally, after cleaning it up a little bit, we got something that's a pretty good approximation to a minimal genome, JCVI Syn 3.0. And here in a picture made by Suzuki is that genome. So the genome, as I said, is built in 8 segments. And these are made by reduction of the same regions in the original mycoides genome. So the result now, JCVI 3.0, is about half the size of the starting genome. And it grows well. So we're going to spend the rest of the time talking about the analysis of this system. So what functions does it encode? Well, about 230 can be very confidently assigned based on matches to hidden Markov models of that have a member that is really well characterized biochemically. And actually, the equivalogs, the probables, and the putatives are we're pretty confident about these. And, however, surprisingly, there were 149 cells, genes which we classified either as generic or unknown. 65 that we had no idea what they do, and 84 that are, for example, we might know this is a transporter, but we don't know what it transports. That's what the kind of thing I mean by generic. So when these are blasted against a panel of organisms ranging from other mycoplasmas to archaea with other prokaryotes and eukaryotes included, then we get this rainbow diagram, which was made by Chuck Merriman. And so many of the equivalogs match things throughout all life, whereas as we go to the less certain classes, we have more and more genes that are specific to the mycoplasmas. All the genes are real, by the way, because this matches here in the bottom line are to non-mycoides mycoplasmas. So they are all reading frames that are conserved among the mycoplasmas. So these genes can be functionally classified into a number of classes. So they're about half of the genes, 41% for expression of genome information and 7% for preservation of the genome information. About half of these genes are related to the central dogma. DNA goes to RNA, goes to protein, and the other half are related to other cellular functions. As I said, it was quite shocking that when we did this, about a third of the genes did not have well-defined functions. And we've been interested in trying to clean that up, and we've made some progress. So since the publication of SYN3, about a third of those genes have been promoted to generic, from generic or unknown to putative, probable, or equivalogs. And so now there's still about 100 genes that don't have really well-defined functions. But nonetheless, people are very interested in this, and we've been interested in collaborating with a bunch of people who want to study this minimal cell. This is from the Kevin Costner movie. Okay, so here's some of our collaborators, and they include people that we're working with actively now and people who've expressed interest and we're not certain about the status of things. And this list has grown even more over time. And so we're gonna focus, I'm gonna focus the remaining few minutes on cells, studies that have been done in two studies, one on microfluidic studies and studies of giant cells produced by our reduced genome, and another collaboration with a group at the University of Illinois on whole cell computational modeling. So this work, which I'm not actually directly involved in, is done by a collaboration between people at JCVI, Lee Ji-sun and Kim Wise, and people at MIT, James Pelletier and Elizabeth Strakowski, who is at the National Institute for Standards and Technology. So when we first looked at SYN3, Mark Ellisman and Tom Deering at UCSD looked at the cells and saw there were giant cells. So this is a typical cell, but here this cell has about an order of magnitude bigger volume. It's a little more than twice the diameter. It's a sphere approximately. So we found that these giant cells came from the fact that there were a number of genes removed from segment 6. This shows cells produced from transplanting 8 different genomes. Each one has a single reduced segment, and the other 7/8 are wild type. And segment 6 reduced produces lots of these giant cells. Here's some other pictures of this. These are typical cells for the mycoides, and this is the giant cell. These, you'll notice their pictures I've shown are done by various techniques, so they have different appearances. Okay, so James Pelletier looked at these cells in little chambers where he could look at what was going on in with single cells. So if we look at this for one particular chamber, just to show kind of result, there's a cell, it starts, this is growing and it's blowing up and making these huge vesicles. This is staining with M. cherry for total cytoplasmic content. So we produce lots of vesicles, you see there. And this is very atypical. So we were interested in getting a cell that was better behaved, like mycoplasma should be. And we added back a segment of 19 genes that restored the normal morphology. And on further dissection, these 7 genes indicated by the red arrows are the ones that are important. Some of these are cells we knew from their annotation are involved in cell division, SEPF and FTSZ, and others are not. And we don't know now even what their function is in this process. And so control of cell division is an interesting thing that can be approached with this system. JCVI SYN 3.0 has a morphology similar to bacterial L forms, and adding 7 genes to it results in a normal, well-behaved cells, suggesting SYN 3 and SYN 3A, which is a construction that has those cells plus others, may be a useful model. I see Jacqueline, do I have a limited
You have
time?
about 5 to 6 more minutes left before we'll take questions.
Okay.
Thank you.
So I'm just going to, we're interested in the modeling of the cell also. And this is work with the Luthi-Schulten group at the University of Illinois. And one thing we've done is to take the metabolism of the cell and construct a metabolic model using flux balance analysis. And this is a mathematical model that's not very computer intensive and doesn't require a lot of kinetic data. And so it can be used to estimate growth rates under assumptions of like steady state condition, and it relies on a set of equations that just rely on mass balance. So we've constructed a metabolic model, and almost all of the reactions in this are supported by strong annotation of the genes, and the model includes 336 reactions with 155 genes and 300 or so metabolites. And it reduces the experimental doubling time quite well. And this group is interested in a number of other things that we don't have time to get into. One is kinetic modeling, when they're just producing a kinetic model of the genetic information processes in the minimal cell. And they've also been interested in 3D models, and in particular in all atom molecular dynamic models, and modeled 2 billion atom construction that has a membrane with water inside. So we're interested in the whole cell model to identify limits of our knowledge, suggest experiments, and we have the hope that if we can build an accurate, comprehensive model that's predictive, it means we can understand the cell in terms of physics and chemistry, essentially. So we have overall tasks for the living cell is to reproduce itself, and there's no unique set of genes for this. There are biochemical functions that require one or more functions, and we propose there's a minimal set of tasks. For example, here are some tasks involved in translation, and one of the things you have to do to get translation of a protein is acquire all of the amino acids. So to acquire lysine, this can be done in different ways. There are different pathways that can synthesize lysine. And on the other hand, we can transport lysine in from the medium, which is what our minimal cell does. And we've begun the process of trying to make a more complete list of all the tasks that are required by a minimal cell. And so the minimal cell idea seems simple at first glance, but it's more complicated than it seems. And we have a tool here to investigate the first principles of life, and we're developing organisms we're developing organisms for use in further studies. Okay, our vision is a cell where it hasn't changed. We aim to make a predictive model and describe the cell in terms of a set of universally required tasks. And And of course, by thanking, this is a list of many of the people who have been involved in this progress at the Institute at JCVI and with our collaborators. Thank you.
Great, thank you very much, Dr. Hutchinson. We have quite a few questions from the audience, so we'll go ahead and address those now. The first comes from Frank Lee. He asks, what environment are you targeting your minimal synthetic cells to be able to survive or thrive in? For example, a nutrient-poor medium like 0.1 micron filtered boiled seawater in sunlight at 15 degrees C, or a rich medium like LB at 37 degrees C.
Yeah, am I on? Yes, we are trying to make a cell that will grow in, we want a minimal cell, so we want a cell that will grow in the richest medium we can provide. So we're trying to make it as independent of synthetic metabolism as possible to get down to the bare minimum we can require. So there's serum and stuff that, undefined things in our typical medium. We have a partially defined medium now, though, that's interesting for these questions. Thank you.
Okay. Sure. The next question from Gabriel Lopez. Can the amount of genome reduction be correlated to the increase in growth rate? Or is there a particular gene set that was eliminated that resulted in the greater than 5 times increase in growth rate?
Okay. Yeah, that slide may have been meaning that greater than 5 times. It says that the minimal cell we made is smaller and grows 5 times as fast as another species of microplasma, which has the smallest genome of any known naturally occurring organism. So there isn't an increase. In fact, there's a reduction on deleting things from the mycoides genome.
Okay, thank you. Anha Ehrman asks, what are the current approaches to investigate the function of the unknown genes in the minimal genome?
Well, one of our main functions has been to follow the literature and look for new information about genes that have good matches to our genes. There are a number of things that could be done. I mean, for example, one can imagine there are a lot of transporters with undefined substrates. One can imagine setting out to study a group of things like that in particular, for example.
Good. Felipe Quiroz asks, the working definition of the minimal cell focuses on number of genes. The progress is remarkable. He's curious to learn about what your team has learned regarding the associated regulatory systems or sequences.
Well, we see promoters and things. We haven't specifically studied regulation much. We think this is not necessarily a highly regulated system, and it may not be of great interest for studying regulatory questions. I could be wrong about that, though.
Okay, and the last question for today from Joanna Xavier. Do you see implications of your work in the question of the origin of the first cells? Specifically, if there is an empirical minimal number of genes or tasks for a cell to live, what possible simpler transition states could have existed at the origin of cells?
Yeah, I don't know if our system is good for looking for the origins. I think it may be, I mean, things like the cells that replicate without forming, the replicate and form these vesicles, and the division is not well regulated. That could represent a state that occurred in a primitive organism before the current cell division mechanisms have fully evolved. And so it might be a useful system for studying that kind of question.
Great. Okay, thank you very much for your talk today, Dr. Hutchinson. Okay, we are going to transition over now. So our next